Today, we're diving deep into a really significant announcement, the 2025 Nobel Prize in Physiology or Medicine. It was just announced this past Monday, October 6th. And this prize, it recognizes research tackling, well, one of biology's toughest balancing acts. It's all about how we keep our own immune system under control. The winners are American scientists Mary Brunkow and Fred Ramsdell, and also Shimon Sakaguchi from Japan. Their work is credited with really creating huge openings for new treatments. And interestingly, in two very different areas, cancer and autoimmune diseases.
Yeah, it's an incredibly important area. The prize highlights their work on something called peripheral immune tolerance. You can think of it like this. The immune system has to walk this tightrope, right? It needs to be aggressive, fighting off all the microbes and viruses we encounter daily. But at the same time, it absolutely cannot turn against the body itself. So this prize, it honors the team that really uncovered the key mechanism keeping everything in balance, preventing that self-attack.
Peripheral immune tolerance. It sounds, well, technical, but it really gets to the heart of the breakthrough, doesn't it? It's literally the body managing self-defense without accidentally causing self-harm.
Exactly. And that word peripheral is actually quite important, often gets overlooked. So T cells, the immune cells, they get some initial training in central places, like the thymus. That's like checkpoint 1.
Right.
But sometimes a T cell might slip through that training. It gets out into the body, the periphery, and mistakenly sees a healthy cell as an enemy.
Ah, so that's where the peripheral part comes in.
Precisely. This peripheral tolerance mechanism acts as a second checkpoint, a crucial one. It has to shut down those rogue cells before they can cause autoimmune disease.
Right, like a two-factor authentication for the immune system. So Brunkow, Ramsdell, and Sakaguchi, they identified the specific cells doing this second check.
They did. They really brought this specific group of white blood cells into the spotlight. They called them regulatory T cells, or Tregs for short. The Karolinska Institute, the body that awards the Nobel, they used a great analogy, called them the immune system's security guards.
That's a good way to put it, because they're not the attackers themselves, are they? They're more like mediators, peacekeepers.
Exactly that. Mary Brunkow pointed out that even though these Tregs are pretty rare, just a tiny fraction of our T cells, they are incredibly powerful. They act as the system's internal braking system or dampening mechanism. Their whole job is to rein in an immune response, stop it from going overboard and attacking healthy tissue.
Okay, but if they're so rare, finding them, studying them, it must have been incredibly difficult, right? Never mind using them for therapies. How did they even pinpoint these specific cells?
Yeah, that was the challenge. They needed a unique identifier, a cellular ID card. And the key breakthrough, the real linchpin, was identifying the specific gene that controls these cells, the FOXP3 gene.
FOXP?
Yes. Brunkow, Ramsdell, and their colleagues, they isolated this gene, and that allowed them, really for the first time, to reliably identify and track these vital regulatory T cells.
I recall reading that Ramsdell initially found FOXP3 in mice. Published that back in 2001, I think.
That's right. The initial discovery was in mice.
Well, why was just identifying this gene so much more significant than simply having a marker to track the cells?
Well, because it turned out FOXP3 wasn't just a marker. It was essentially the master switch for self-tolerance. What made this discovery hit so hard so immediately was the simultaneous finding that mutations in this exact gene, FOXP3, directly cause a devastating autoimmune disease in humans.
Oh, wow.
Yeah, a condition called IPEX syndrome. When that FOXP3 master switch is broken, peripheral tolerance just collapses. It often leads to death in infancy.
So it proved that the gene wasn't just correlated, it was causal.
Absolutely. Jeffrey Bluestone, who worked with Ramsdell, summed it up perfectly. He called these Tregs the master regulators of the tolerance of the immune system. Once you find the regulator that causes total system failure when it breaks, well, then you have the key to potentially controlling the whole process.
And that control, I imagine, is what everyone is racing towards now. If Tregs are the master regulators and FOXP3 is the switch, what does that mean, practically speaking, for treatments? Are we talking about injecting more Tregs or boosting the ones we have? How does it work?
It really means both, and potentially more. Their work essentially launched this whole field of engineering these Treg cells. For autoimmune diseases, for instance, the idea is to grow and possibly engineer more potent Tregs in the lab, expand their numbers, maybe make them more specific, and then infuse them back into the patient to suppress that misguided immune attack.
That's a huge leap, isn't it? From understanding a mechanism to actually engineering human cells for therapy. Let's take a quick detour, the human side. How did the laureates actually find out they'd won? Those stories are always interesting.
They really are. Mary Brunkow, she's a senior program manager in Seattle now, apparently found out in a
system attack tumors
right, dial them down.
and autoimmune diseases where you want to boost their function to calm the immune system down.
Exactly, dial them up. It's about controlling that regulatory dial.
And the scale of this effort is, well, it's staggering. The Karolinska Institute mentioned that even though specific therapies aren't approved for market yet, there are more than 200 human trials involving regulatory T cells happening right now, globally.
Yeah, 200. That's a huge number. It signals enormous commercial confidence, you know? A full-blown race is underway. And the big pharmaceutical players, they're placing massive bets on who can successfully engineer these immune regulators first.
Can you give some examples?
Sure. We mentioned Sonoma Biotherapeutics, where Ramsdell advises. They're backed by Regeneron, the big U.S. drug maker. They're focusing on things like inflammatory bowel disease.
Okay.
Then you've got companies like Quell Therapeutics. They've partnered with AstraZeneca, also targeting IBD and other major autoimmune conditions. And other giants like Bayer, through its Blue Rock subsidiary, they're actively exploring the same Treg approach too.
So it's not just academic anymore. This is big business, multi-billion-dollar competition to control that second immune checkpoint.
Absolutely. The race is definitely on.
Before we wrap up, let's just quickly
Global pharma giants are already running. So it makes you wonder, What stands out more right now? Is it the sheer elegance and importance of that initial scientific discovery, the finding of FOXP3 of the Tregs? Or is it the tangible promise, the potential application we're seeing unfold, the hope that, as Sakaguchi put it, cancer might become no longer a scary disease? It's sort of a fascinating question about how we value science. Is it the initial spark of the idea, or is it the proven fire of clinical application? Something to think about.